All right, in this video lecture, I am going to be looking at Mendel's principles and talk a little bit about probability and how we can use Punnett squares to predict possible outcomes in crossing. Then I'm going to throw some more vocab terms at you and really dive into alleles and how we can describe alleles. So going back to Gregor Mendel, the Austrian monk who did experiments on pea plants. The thing about Gregor Mendel is that he counted everything. So when he crossed, two true breeding organisms for a certain trait, and then the offspring were produced, he counted and recorded what those offspring looked like, and he had notebooks upon notebooks upon notebooks of his data. And so he had plenty of data to analyze, and that's when he started to see that in the second generation, there was that ratio of 3:1, where that recessive trait that disappeared in the first generation reappeared in that second generation. And because Gregor Mendel had a pretty strong mathematical background, he actually was able to use probability to explain his results. And so what is probability? That's just the likelihood of an event that will occur. So if you flip a coin, what is the probability of a heads outcome? So a coin only has two sides. It's heads or tails, and so heads 1 out of 2, that'd be 50%. Now, let's say you wanted to do 3 coin flips in a row, and you wanted to figure out what the probability would be to get heads each time. Again, you can just do math to figure this out. And what you do, let me get my pen, is you multiply the probability for one outcome, and then you kind of set it up like this. So the probability of one outcome of getting heads is 1 out of 2. But that's for the first coin flip. That's supposed to be a multiplication sign. But then for the second coin flip, it's still 1 out of 2. Okay, the whatever happens here does not affect here. These are independent events. And then we said, okay, we want to know what's the probability of getting heads 3 times in a row. So 1 times 1 times 1 is 1. And then 2 times 2 times 2 is 8. So the probability of flipping a coin and getting heads 3 times in a row is 1/8. Okay, each flip is an independent event. It's not impacted on prior. Like for example, if a couple, they were going to have a child. And there's a 50% chance that it would be a girl and a 50% chance that it would be a boy. Well, if they get a girl the first time, that doesn't mean that a boy will occur the next time. So that's what I mean by it's an independent event. It's not affected by the previous event. So here's just a snapshot of Gregor Mendel's data, looking at the 7 either-or traits. You're either purple or white, yellow or green, round or wrinkled, green pod or yellow pod. And this is just some of the data that he has collected. And again, look at that ratio, how very close this first number is to 3:1. So 3 dominant to 1 recessive. So how can we explain these results? Well, when alleles of the parents, okay, each parent has 2 alleles for a gene, and when gametes are made, gametes are sex cells, sperm or egg, those alleles segregate or separate randomly during the formation. So what if I had a question that is, what is the probability of an offspring with 2 recessive alleles if the parents are big G or little g? Well, again, that means this is what the parents look like. Big G, little g. Big G, little g. All right. So what are the chances of 2 recessive alleles, right? So there's 2 letters, and they carry 1, so it's 1/2. And the other parent, they carry both alleles, so again, it's 1/2. So we multiply across, and there's a probability that the offspring, 25% of them would be 2 little g's, or that it would be little g, little g. Let's say the gametes, let's say we have a parent that is 2 little g's. I decided I'm going to go 2 little g's. And you have one parent that's big G, little g. And they said, hey, what's the probability of an offspring with 2 recessive alleles? So looking at this first parent, it's 2 out of 2. Both alleles are little g. 2 out of 2, 100% chance that the gamete from this parent is going to be a little g. And then the other parent, it's 1 out of 2. Okay, that kind of looks like a plus sign. That's a multiplication sign. So I multiply across. It's 2, 4, and that's 50%. Okay, 50% chance that the offspring would be little g, little g. I'm going to try to remind myself to come back to this slide. So ways that you can describe alleles is homozygous or heterozygous, and homo means same, and we are looking at the alleles here. So when I say that a homozygous dominant individual, okay, homozygous means the letters are going to be the same, and then because I said dominant, well, that means it's going to be an uppercase letter or capital letter. If I said homozygous recessive, we went over a dominant versus recessive in a previous lecture, and so recessive is 2 lowercase letters. Homo, same, zygous, recessive, little or lowercase letters. Heterozygous means that you carry both alleles, capital and lowercase. Hetero means different, that the alleles are different. So here you can take a look at, this is a chromosome, and this is a gene. And in this case, this chromosome's carrying the dominant allele for that gene, and same with this chromosome. So we would describe it as homozygous dominant, big B, big B. These 2 chromosomes in the middle, again, we're still looking at the same gene, but you can see that they have different alleles, big B and a little b, and so they are heterozygous. And then over here, we're still looking at the same gene, but a different form of that gene. They carry the recessive allele, so little b, homozygous. Another way that, when we talk about genes and their alleles, we have to talk about phenotype versus genotype. And so phenotype means what is the trait that you can see in that offspring, okay, the observable trait. So when you look at, the color of the pod, is it green or yellow? The color of the seed, is it green or yellow? Is it wrinkled? Is it round? So you can say seed shape. Those are examples of phenotype. So in humans, eye color would be the phenotype. That's the observable trait. You can look at someone and, yep, they have blue eyes. Yep, they have brown eyes, or, hazel, and so on and so forth. Genotype means you are looking at the alleles, and the alleles are basically the letters, okay? So let's say that green pods are dominant over yellow. Okay, so green is dominant, so I know they have a big G, but I really don't know what the second allele would be. But genotype means you're looking at the letters. So here we have flies with their wing shape, and it says here normal wings. That's the phenotype. You can look at this fruit fly, and you can see that they have normal wings. So we're looking at wing shape here. And what's going on genotypically, what we can't see, the letters, in this case, they're homozygous dominant for normal wings. These next 2 flies, you can see that they have normal wings, but notice that you can't tell if they're homozygous or heterozygous, but in this diagram, they show you that, yes, they are heterozygous for normal wings. And then homozygous wrinkled, or homozygous recessive, and with the recessive trait, the only way you can have that recessive trait is if both alleles are lowercase. So I mentioned how I don't have a widow's peak, but my husband does, my children do. And no widow's peak is recessive. And if I were to use W for my alleles, I would be little w on both of my pairs of chromosomes, okay? So that means that I can only give a little w to my children, right? And my husband has widow's peak, but I don't know if he's heterozygous or homozygous. You look at him, you can see the widow's peak, but I don't know if he's heterozygous or homozygous. Now, both of my kids have widow's peak, so that means they carry the big W, but then I can only give the little w, so I know for a fact that my children are heterozygous. So just one more way to look at phenotype. The trait that you can physically see, okay? And so this is Gregor Mendel's experiment, 2 true breeding traits. And then here's my first generation showing the dominant trait, and they're hybrids because they carry that recessive allele. And then in the second, after he allowed them to self-fertilize, there's that 3-to-1 ratio. Here's what's going on genotypically with the letters. Big Y, little y, 2 true breeding plants for their traits. You can see that with the F1 generation, they're hybrids, big Y, little y. And then what's going on in the second generation? Well, yes, you have 3-to-1 for big Y to little y, but then notice that 25% of them are purebred, and 50% of them are hybrid. So one way to show the outcome, the probability of an outcome in cross-ins is to use a Punnett square. And we are just going to focus on one factor trait Punnett squares, and we'll worry about dihybrid or two cross-ins in a different lecture. But we call these monohybrid when you are looking at one trait. And so the steps is you start with the parents. And so you look at the parents' genotypes, or you try to figure out the parent genotypes. Now here I'm giving you the letters, okay? But if I said a homozygous dominant individual is crossed with a heterozygous dominant individual, well, you need to know what homozygous and heterozygous means. Homo means the letters would be the same, and then if it's dominant, they're going to be big letters. If it's homozygous recessive, they will be small letters. If they say heterozygous, boom, it's already big letter, small letter. So we're going to say big B. I didn't tell you what the phenotypes are here. Big B is going to be the trait for brown eyes. And, wow, and a little b is for the blue allele, for blue eyes. So the only way you can have blue eyes is if you are two little b's. So here in this first Punnett square, you start with the parents. I'm going to put parent 1 at the top. So big B, and then you stick the other parent off to the side. And it's kind of like a multiplication table. You know what? I'm actually going to use a different color just so you can see how the alleles go across. So this might take me a little bit longer, but since I can't point, really. Okay, so what you want to do is you're going to fill in the square. And in order to fill in the first square, you got to take a look at the letters that are at the top and off to the side. So this little b, you know, fills in this box and this box. And then this big B, you know, put one here and here. All right, and then we're going to move on. Okay, so this big B, I'm going to put a big B here and here. And when you fill in your Punnett square, you always want to write with your capital letters first. So there's the little b, I go across. Okay, so I'm going to use a different pen here to show you that little b went there, and this little b went there, and that little b, or that big B went down here, and here, and here, and here. Okay, so that's kind of how I filled in my Punnett square. And now I might ask you, what is the genotypic ratio, right? Because if, you know, as a good science teacher, I should. Genotypic ratio. So what that means is you take a look at the letter combinations in your Punnett square, and I only have one letter combination. It's big B, little b. So I'd write big B, little b, and I say how many squares out of this Punnett square are big B, little b, and it's 4 out of 4. Now I might ask for a phenotypic ratio. Phenotypic means the trait that you can physically see. So I said big B is dominant over little b for blue eyes. So brown is dominant over blue. Well, all of these squares contain the big B. So that means that they will be all brown. And so it'd be brown 4 out of 4. Okay. All right. So I'm going to shrink this. So that's for the first one here. Okay. So let's do the second one here. Parent number 1, you know what? I'm going to switch this up. Let's change this to a little b. So parent number 1, big B, little b. Parent number 2, big B, little b. All right, so now I'm going to fill in. So this big B goes here and here. And I'm going to switch my pen. And the big B goes here. Here, so you always write your capital letters first. This little b, whoops, see, I already messed up. Go me. Slide this over. All right, so I put a little b here, a little b here, and then got one more row to fill. 2 little b's. All right, so again, to show you how those letters or how these squares were filled in. Big B there. Big B there. Little b, little b, little b. Okay, so now let's figure out our genotypic and phenotypic ratio. All right, so erase this. This is going to be different. Genotypic. So I take a look at my Punnett Square, and I can see I have multiple genotypic ratios. And the best place to start is just pick one. All right, so I got big B, big B. How many squares are big B, big B? Well, there's one. Looks like it's just 1 out of 4. Okay. Big B, little b. How many squares are big B, little b? So 1, 2. 2 out of 4. And then my last genotypic ratio there. Little b, little b. How many squares are little b, little b? And there's just 1. Notice that it all adds up to 4. But my genotypic ratio is 1 to 2 to 1. All right, phenotype. How many are brown? So brown is dominant over blue. That means brown, we're looking for big B. So any square that has a big B is going to be brown. So this has a big B, there's 1. This has a big B, there's 2. This has a big B, there's 3. This one does not. So I'd go 3 out of 4. All right, so now blue, the only way you can show the recessive trait is to be homozygous recessive for it. So I'm looking for 2 little b's, and there is 1 square there, 1 out of 4. So that's going to be your assignment. The back page is all about Punnett squares. Now I'm going to go back to this slide right here. Notice how I used math to figure out probability. I didn't have to make a Punnett square, but you know what? Just for the heck of it, let's just, now that we know a little bit about Punnett squares, make a Punnett square to make sure that my math was right. So here's parent 1, big G, little g, big G, little g. And when I fill this in, this is going to be 2 big G's. This is going to be big G, little g, big G, little g, and then 2 little g's. And so they wanted to know what's the offspring, what's the probability of an offspring that is little g, little g. Well, I look at my Punnett square, look, 1 out of 4. All right, this combination over here. Okay, so parent number 1 is 2 little g's, and the second parent is big G, little g. So I fill this in, big G, little g, big G, little g, and then it's going to be little g, little g, little g, little g. All right, so how many squares are little g, little g? They're right here on the bottom, 2 out of 4. I can change this. Instead of saying 2 recessive, maybe I want to say 2 dominant, right? So the math here, okay, there's a 50% chance that they could be big, that the gamete or the zygote or whatever, the offspring will get a big G from parent number 1. And over here, again, 50% chance, I multiply across, 1 out of 4. And I look at my Punnett square, boom, 1 out of 4. Over here, let's just see if this works here. 2 dominant alleles for this crossing. So this first parent, it's 0 out of 2, right? And then the second parent, it's 1 out of 2. And when I multiply across, 0 times 1 is 0. Notice that in this Punnett square, there are no dominant alleles. All right, so that does it for Punnett squares. You do have a short assignment on 12.2 part 1. And then the next day, we will do a Punnett square packet just to get Punnett squares down.